Welcome everyone to the last MVZ lunch of the semester. Carly, is that correct? Thank you. And I want to remind everyone that we're going to have our MVZ symposium in January and the week before classes start. And we'll be sending out more information about that in the near future. And so I want to wish everyone a safe end of the semester, and I will turn it over to Chris to introduce our speaker today.
Oh, great. That was fast. Okay, so it's my great pleasure today to host Prosanta Chakrabarty, who is a professor and curator of ichthyology at Louisiana State University, currently on sabbatical in Ottawa. So Prosanta did his bachelor's at McGill and then his PhD at University of Michigan with Bill Fink, and a postdoc at the American Museum of Natural History with John Sparks and Melanie Stiasny, I think mostly working with those two, and then started his faculty position at LSU. And I think Prosanta, I think, is essentially living the dream of what every child imagines it's like to be a museum curator and to be an ichthyologist. This is Prosanta. I mean, he's done fieldwork in over 30 countries now. Most recently, I mean, Thailand, Haiti, I think Sri Lanka, Panama, sort of more, in the past. I mean, at any given time, Prosanta is off in the caves of Madagascar, spelunking and discovering new cave fishes, or the caves of Thailand looking at walking angelfish that we'll hear about today. Or he's off studying deep sea, bioluminescent ponyfishes, or describing, new species of cichlids all over the neotropics and old world. And so, his career epitomizes discovery and what we're all fascinated by in terms of museum collections and developing collections. He is also has not given one TED Talk, but 2 TED Talks, which I highly recommend, reaching an audience of over 5 million people now. So he has an incredible outreach and ability to engage with the general public. He's been elected to American Association for the Advancement of Sciences a couple of years ago now. He was a program officer at NSF. He's secretary of Ix and Herps. I could go on. The list goes on and on, but I'll hand it off to you, Prosanta. Take it away. It's my pleasure to have you here today, and I hope that eventually we'll be able to have you here in person too.
Thanks so much. That was quite an introduction. That makes me feel a lot better about my career than I usually feel about my career. So thank you. Let me share my screen here while I think I can do this. So let's see. So can everybody see that? Yeah, thumbs up. Okay. So Chris asked me to talk about fieldwork, and I know this is your last seminar, and I've given a bunch of these virtual seminars now, and the one thing I've learned is, like, no outlines, just go for it. Let's ride a roller coaster and see what kind of theme we can get going. But I'm glad he asked about the fieldwork, because it kind of gives me these interstitials between subjects. So I do hope to make it fun, but also talk about some pretty serious topics as well. And when I think about fieldwork, it's not just the animals, I think about the people. And so I actually want to start, this is a picture of me on the Ottawa River a couple of weeks ago. And I'm in Ottawa, but it's also Algonquin, Anishinaabe territory. This is unceded land. And thinking about that land acknowledgement and the people that I've been with in the field, it's their land. And one of my earliest papers was on sturgeon, but this is the first time I held a living sturgeon. And I was with a fisher, and it was clear to me, within 10 minutes that she knew much more about sturgeon than I did. She knew about population levels. She knew about how to collect them. This guy was released unharmed, but I was learning a lot more from her as a fisher who knows this land better than I could have told her about from my papers. So this talk will be about fieldwork, but also about the people behind it. I also want to acknowledge, last month was a terrible month for women in STEM in terms of how perceptions and how people were related to these two publications. So the first one took a lot of heat because it basically discussed the price women pay for mentoring other women, and it got a lot of heat, and deservedly so. But it also was a call, I think, for men to step up as mentors, and to acknowledge our own female mentors. So Chris mentioned one of mine, Melanie Stiasny, who was actually my undergrad mentor, and got me into fishes, and I wouldn't be studying ichthyology without her. But also Ellen Pickett, who I worked with at Wildlife Conservation Society. And so we have to take that paper in particular with a grain of salt. This other one, McKinnon and O'Connell, actually came out at the end of November, and it brought up a different point. So this is a word cloud about how women are perceived for doing the kinds of things that I get a lot of credit for. So I do a lot of science communication, I do a lot of outreach, and I never get called any of these things. And so just acknowledging how differently people are perceived for doing the kinds of things that some of us get credit for. And again, a call for us men to step up a little bit. So this is me in the field with a man named Shay in Tanzania. And this is a mangrove habitat that goes up about 9 feet. And Shay had lived there his whole life. And he doesn't know the scientific names of some of these gobies that I'm collecting for LSU and for the local collections. But he, again, just knew so much about this landscape and was teaching us about the mangroves and what this area means, not just in terms of the fishes, but what it means in terms of his livelihood on a daily basis. So again, we'll be talking about the individuals in these collections, and I'll be using these videos just to break between subjects because Chris gave me permission to do so. But keeping on that theme of science communication and the need for it, Aristotle's often given credit in the West for inventing science, but he also invented science communication. So he sort of parsed out his work into the esoteric, which is what we're doing when we discuss papers at a conference or just writing those papers for other scientists, but also the exoteric with an X. And that was his work to talk to a broader audience. And he saw these as very important for all of his work, to talk to the public and explain to them in a way that you don't get if you just do the esoteric. And I've heard once somebody say, if you put the average scientist in front of a crowd, they might do more harm than good. But there is a point where we need to think of the broader impacts, but also the communication we do with the general public, especially I think now in the age of COVID, and I'll discuss that a little bit further. So we as scientists, we're building this truth, right? We're testing hypotheses, we're discovering things, but people won't believe that truth unless they trust you. And that trust has to be earned. And so, if somebody, like the President of the United States, he says the science doesn't know, which he said, we have to counteract that to, get people to trust us as well as scientists. And so to get to our truth, they have to trust us. To earn that trust, I think we have to explain things in a way that's manageable and it can be understood. So, with the genomic era and people sending their spit and money to 23andMe and other companies to learn about their ancestry, I think we have to get ahead of how that is often interpreted. On the left here, we have white supremacists drinking milk to show that they have the lactase mutation that shows that they have Aryan ancestry, missing the point that West Africans also have that mutation. Why they need to drink milk without shirts on in cold weather, that might be part of the Neanderthal genes that they also have, but their interpretations need to be explained by science, right? And both sides of the political spectrum can get this wrong. So Elizabeth Warren used a DNA ancestry test to show that she had Native American ancestry, but because of recombination, she may have had zero evidence of that ancestry. And there are people with even more tribal affiliations and connections that might show zero Native American ancestry, but even if they had it, right? Just as the complexities of recombination erasing part of that history. So along those lines, these genome-wide association studies, which are great. There's a lot of data there. It's information that we need. But when we create these studies, there needs to be some communication as well. Eugenicists, including scientists like David Starr Jordan, called for sterilization, elimination of people who were alcoholics or poor or gay. And so when these studies come out, they need to come with some science communication as well. And it comes best from the scientists themselves. This same-sex behavior one, I think, did a really good job at attempting to discuss the topic of finding genomic markers for same-sex behavior. Other studies maybe haven't done such a great job. But even when they do a great job, like that Ghana et al. Paper, there's still an app that was produced, the How Gay Are You app, which was purporting to show how your genetic markers may influence your same-sex behavior. Of course, it can't. That paper itself discussed how they found sort of tenuous connections between any marker. And so this app has seemingly very nefarious purposes, especially because it was developed out of Uganda, a country that criminalizes same-sex behavior. So even when we have the best intentions, it can have these terrible consequences. I don't know if it's helpful to anyone, but one of the tools I use for explaining to a broad crowd, how to avoid or how to discuss homophobia is that very few people actually have their karyotypes done. So there are few genes that relate to secondary sex characters that make someone male or female. So the SRY gene that can be on the XY, that is when you learn in high school biology that makes you male, that gene may have actually moved to the X. So you might be an XX female and an XY female, and very few people actually know if they are or not. So I have them consider how would your mind change if you actually were an XY male, female, or an XX male, if that makes sense. So these are tools of science that we can use to explain genomics. These are twin girls, or twin women now, that are probably gonna check off different boxes for race. So just because the few genes for hair color and eye color and skin color are expressing differently, but they have the same parents born at the same time. So explaining that race and gender and sex and sexuality are things on a spectrum isn't something that I learned when I was learning evolution as a grad student or before. And so I think we need to get in front of this information and explain it to as many people as we can as scientists in order for people to trust us. This includes telling stories and explaining people of the past that may have been largely forgotten. So I'm working on a book, and this is a drawing that Ethan Kokak did for me about John Edmonstone, somebody I hadn't heard about until pretty recently. But he was the person that taught Darwin taxidermy before he went on the Beagle and before he even signed up for the Beagle. And he was an enslaved man from South America. So his influence on Darwin is pretty important to show if we want to diversify science. So for me, that was an important story to tell to students to get them interested in evolutionary biology, which doesn't showcase many people like Edmonstone in the past. Same with Marie Tharp. When we talk about Alfred Wegener and continental drift, how often do we talk about Tharp's work? She was the first to map out the ocean seafloor and show the evidence for continental drift that was missing for so long. And when she first found that evidence, it was called girl talk. right? So people didn't believe her. She wasn't allowed on the ships to map the seafloor. She had to work with the data itself. And Jacques Cousteau was trying to prove her wrong and ended up proving her right. So these stories matter. And so I just wanted to sort of start this talk by acknowledging those forgotten people and our need to explain the exoteric side of science. So maybe I feel lucky that I work at Natural History Museum so that I can explain topics like evolution and genomics pretty, fairly easier. It's like a gateway drug to these more complicated topics, especially when, we have to deal with fake news, which has been a prominent topic over the last 4 years, right? But working at a natural history museum, each of these specimens is not just a data point or data points, but a story. So it's a great opportunity to talk about other work. And I'm glad that MVZ now has a real, vertebrate curator and the fish curator. Before you were a tetrapod museum, I'm sure you've heard that before. So I was ecstatic to see you guys hiring Chris. But one of the fun parts of being at a museum, as many of you know, is when we describe species, that usually gets a lot of attention because people understand what a new species is, or they often do. And so these are the 15 species I described, and that's been an important showcase of my work, but what's behind that doesn't get as much attention, which is the taxonomy. So the revisionary work that also goes along with naming parts of the tree of life. So many of us do this, but it's a harder thing to explain than what a new species is, right? So if I name a new genus, that doesn't resonate as well. But we have this great opportunity with COVID because COVID has multiple names, right? COVID's the disease, SARS-CoV-2 is the virus. And there's evolutionary explanations of why we call it SARS-CoV. It did start off as the novel Wuhan coronavirus cluster, right? And we had to change the name and the taxonomy. Maybe more than one of you is probably going on to NextStrain to just look maybe morbidly at the evolution of this virus as it mutates. But this is a phylogenetic tree, so it's a great opportunity for us evolutionary biologists to use this tool that's been used for explaining this virus and how it works. For instance, that the New York strains initially were from Europe and not from Asia, right? So we can see the mutations over time. And so we can use that tool to explain what we do. And this is an example of how I use it. So this is a tree of Central American cichlids with a fossil that I redescribed from Haiti and changed the genus, and it changed the date of when cichlids arrived in Central America and the Caribbean, and it changed the dates across the tree. So that's a much easier thing to understand for the general public if we couch it within the context of COVID, or at least I think so. The tree of life for many people will be this strange abstraction, even if we're in that tree. So whenever we have tools like the phylogeny of SARS-CoV-2, I think we should use it. The same is true of global warming. So, people were generally on board with it occurring, but it became fake news, right? And so much fewer people believe it now because they trust the people that say it's fake news instead of trusting the people that say it's something that we have to worry about. And why do they trust those people? The other side has better stories, right? So I think part of what we need to do is explain not just sea level rise and temperature changes, but also individual stories. There's a sort of horrible quote, but useful quote by Stalin that's something like one death is a tragedy, a million deaths is a statistic. So if we talk about individual species and what will happen to them, that may help in our quest to explain more difficult topics like climate change. So this is a paper from one of my former students about Chorioheros wesseli, a microendemic. It's found in very few places, but this species will likely go extinct. So I can talk to the general public about this individual species and its impact of sea level rise, not just globally, because individual qualified stories can sometimes be more impactful than just quantified numbers. Ooh, I skipped the video that I'm sure Chris wants to see. I don't know if it's playing. It's strange. Let's see. Yeah, there I am. So people like sort of fancy fieldwork videos and talks, and I'm not even sure why I'm showing this one, except I was stuck in a cave in Western Australia, and yes, we describe new species, but caves are really important. So I study diversification and how it happens, and caves are great places to get stuck in, and species get stuck in caves. And so the isolation that you need for allopatric speciation is really obvious there. And one of the cave fishes that we studied that I think Chris mentioned a little while ago is this cave angel. So this is Cryptotora. In purple here is its pelvic girdle connecting to its vertebral column. It has hips. Fish don't have hips. So this is kind of a neat species. We just got a Understanding Rules of Life grant from NSF to look at this anatomy, to look at the biomechanics and the phylogenetics. And we actually got to go there. So just before COVID hit, or maybe while COVID hit, but we didn't know about it, we were in Thailand earlier this year. And the story about these fish, and these are them here, inspired my grad student, Pam Hart, to get into science. It's important to remember, especially as evolutionary biologists and museum folks, how inspirational some of these oddball taxa can be. So these cave angels climb up waterfalls in caves. They don't have eyes, they don't have any pigmentation, and they're really fantastic. We spent 6 hours in this cave with Thai researchers, and this is Pam looking at her muses. And I want to show you what they do. So look at this video. So if you can see, they swim, they can come out of the water. The water's moving fairly quickly, and again, they can move up waterfalls as well. And then they can come back out and swim. And so the idea behind the grant is that they're convergent with some of the earliest tetrapods that came onto land. And they have the hip structure that is convergent, just like if you think of Tiktaalik or something like that. So seeing that there, and then when we came out of the cave, Brooke Flameng, who's the PI on the grant, she's at the New Jersey Institute of Technology, her lab was able to film this video, and you can see they have it's a salamander. They have a salamander gait. They do this. It even raises its head, which is an unusual trait for a walking fish. So you can see how we see that convergence. And so just this year, Callie Crawford, who's looking for a postdoc, by the way, who's in Brooke's lab and my lab, who put down this UCE phylogeny, we're looking at how this morphology changes across the phylogeny to see if Cryptotora in particular had this unusual morphology, or other members of the loaches that this group belongs to, the balatorids. And we're obviously continuing this work and looking at the biomechanics behind this as well. Hips are sort of an odd thing, almost as bad as knees or shoulders for me. So I think the perfect view of vertebrate anatomy is this thing, right? This teleost advanced fish has nothing to do with, our ancestry as tetrapods. I don't know who doesn't see this as a painful walking, bipedal mess that is walking in humans, or even standing. There's a reason that we're all sitting, right? So for me, for a balatorid to have regained this hip, it's actually an interesting thing. And again, this is not one of those loaches, but a teleost showing the perfect end bone structure of how our bodies should be structured. So again, my driving question for my lab is what's driving diversification of fishes, because it's very different in different parts of the world. There's not a lot of fresh water out there, but about half of all fishes are found in that little 2-3%, a lot less than that actually, of fresh water available, while the other half is in the oceans, which there's a lot more of. But there's not as many isolating mechanisms because there's no geographic barriers, or few of them. So as Chris mentioned, I've been very lucky to have traveled to lots of cool places. It's the hardest part is actually getting permits and finding people to work with, which, you know, you get better at as you go along. And so a lot of my work in the neotropics deals with freshwater and basically looking at how the Amazon basin has been filtered as it moves into Central America and then the Caribbean. And then the Indo-West Pacific work has a lot to do with more bioluminescent fishes and marine fishes. And then I collect cave fishes whenever I can. The neat thing about Central America for me, you know, everybody focuses on the Amazon, but there's almost too many species in the Amazon. And I love the Amazon, but Central America has this like neat, very tangible history. So South America was in splendid isolation for tens of millions of years as it broke off from the other continents and actually left behind the Yucatan Peninsula. And the islands that we think of as the Caribbean islands, or at least the Greater Antilles Caribbean islands, actually formed in the Pacific. And they didn't form in one mass like this, but that's because I made this video. But they did drift in the gap between North and South America. And, you know, did that allow some species to diversify and go from South America to North America a long time ago? That's possible. These land masses broke apart from the mainland Central America about 50 million years ago. And we get nuclear Central America and finally the closure of the Isthmus of Panama sometime between 3 and 8 million years ago, or at least that's the story. And this is just like a plate of glass that's been burst and put together. Different Caribbean islands, Western Cuba and Eastern Hispaniola were once together. There's just a lot of history here geologically. And I always want to know if the fishes match this background. And so what my lab does is go to different Caribbean islands and to Middle America, and we look family by family and species by species, who's related to whom and how that relates to biogeography. And we have lots of papers about all this stuff. And for me, it all boils down to sort of this ugly graph that's too ugly to publish, but it's like the easiest way for me to explain my side of the story of what's happening in Central America. So the closure of the Isthmus of Panama totally changed the world. It changed the climate. It separated the Caribbean and the Atlantic from the Pacific. But there are these periods between 20 and 60 million years ago that some species diversified from Central America to South America. And maybe that corresponds with an early land bridge like the one I showed you in that video. And then there's this more recent one that started about 7 million years ago, which may correspond with the closure of the Isthmus of Panama. For me, even more interestingly, is that there's marine diversification between the Caribbean and the Pacific side up to 1 million years ago. And if the closure happened 3 million years ago, which was what the best dates say, how are Caribbean and Pacific species diversifying across that barrier? Well, for me, that means Central America is not just a land bridge, but a spongy land bridge. I mean, if you've been to Panama, you can see both oceans from some places. And for me, so this is a much more complicated story than the geological history as it's been told so far. When I look at, you know, images of where people have done field work, I think of like the maps of Republican and Democrat states where it's all like either red or blue, right? And it's always much more complicated. Where have you actually been collecting? And, you know, talking about the individual stories again, it's nice to talk about biogeographic units, but it's often more interesting to look at it at a finer scale. And I have a student who I recruited from Guatemala, Diego Elias, who just had a paper in Journal of Biogeography that sort of exemplifies why we need to not just collaborate with people from the parts of the places where we work, but also recruit them into our labs. So Diego, because he's from Guatemala, he just has a different familiarity with the region. And he was able to uncover a new area of endemism that really, you know, because his familiarity with this area and knowing the species and working with them, not just in Guatemala, but as he's come to the US and worked at different institutions. This is not something I would have thought to have done, having spent two weeks or 2 months in these areas. So I have a new student from Cuba who I hope to do the same thing with the Caribbean islands. She just has a different view of these areas and the fauna. So, in talking about fresh waters, we're often talking about individual areas and rivers and lakes, but where are the barriers in the ocean? And that's a different kind of diversification question for me and for the fishes. So a lot of the work I've done in the Indo-West Pacific has to do with these pony fishes. They're not the scale, by the way, they're not this big, but they're very, very common. So they're caught in the millions and maybe even the billions every day. So wherever I've been in this area, they're often ground up for cat food or because they're not very appetizing to look at or to eat. I have eaten them. Believe it or not, these represent different genera. So they don't look very different externally. These are the Lygnathidae, the pony fishes, if I haven't mentioned that yet. But they bioluminesce. And so they're really interesting internally. They have these guanine layers of silvery skin inside their body that actually reflects light. So it's almost like a funhouse mirror when you open them up. And they glow light in different parts of their bodies. So this is my festive Christmas tree, a fish. The males flash in different colors in different parts of the body. And they're co-opting this bacterial light that they host inside something called a light organ. Here's some MRIs showing the diversity there. And the males, these are the patches that the males have in different species, and they're very species-specific patches. And it's a group that, where there's some groups, some individuals in some genera that don't have dimorphism, and others that do. So it's a great test case for studying sexual selection, or we thought. But we've never really been able to show very strong evidence of sexual selection, even though the dimorphism is so striking. And part of that's, there's not a lot of comparative groups, so there's not a lot of statistical power in just one family, even with 60 species. But I started to think...
You recording? Yeah. This is a Flacieres. It is net forest.
So you can hear them clicking. So I wondered, when I got to see these things, in the wild, and it's so important to have boots on the ground and not just study the specimens, but if you can, see them alive. The clicking noise was, another way that they may be communicating. So they may be communicating with light, they may communicate with sound. So maybe that muddles the signal of sexual selection, as does the different ecologies. So they're called ponyfish, which most people are disappointed because they think they're going to be seahorses, but these ponyfish, because of the Roman nose that most of them have, as you can see, it comes out. But they have different ecologies, so some of their mouths come up. It was actually with some Sri Lankan fishermen that I was learning the names that they call them, and I speak a similar language to what they do, and some of the names that they gave them were characters, including having big teeth and being skinny, that I hadn't noticed before. And so they actually had names for all the species that I had scientific names for, and even additional ones. So I even named a genus after some of the names that they provided. So again, where it's worth going into the field when you can, when there's no SARS to get away from. But I always wondered, why are the ponyfishes restricted to the Indian Ocean and maybe this part of can you guys see my pointer? Yeah, so why are they restricted to this part of the world and not into the rest of the Pacific or the Atlantic? And I was reading this Ed Young's I Contain Multitudes, and he talked about Lynn Margulis's symbiogenesis hypothesis, which is often derailed because it's pretty much an anti-Darwinian evolutionary idea, because it's the symbionts driving diversification. And I was thinking, maybe the bacteria are preventing some dispersal into different parts of the ocean, even though the bacteria that they host are found in different oceans. Maybe there's some connection there of preventing diversification or maybe even promoting sexual selection. So what if the bacterial strains are different in different species? So that's a pretty crazy idea. NSF also thought it was too crazy to fund. So, but if somebody, I'd love to go into the field with somebody who's looking at the bacteria and can switch the bacteria among different species to see if there's anything going on there. Again, looking at fresh waters, you know, this is a Nat Geo slide showing the rivers of the world, but this is way overemphasized. You know, most rivers you can't see at this view from this height. So this is the boiling river. And, you know, rivers are great barriers, but this is a river that boils. And I made tea at this river. This is in Peru, Peruvian Amazon, that we went a couple years ago. And it's striking how hot this water is. And it's a clear barrier to most species, not just the fish, which can't go into the boiling water. But when there's rain, it does allow for dispersal across this area. And so this is like an extreme example of how rivers are barriers, but an amazing one for the Amazon. And the Amazon is just an amazing place. There's almost 5,000, 6,000 species associated with South America, most of them Amazonian. But most of them are cichlids or members of this group, the Ostreophysi. So lots of tetras and catfishes, and this group that belongs to the electric eels called the knife fishes. And I always wanted to study this group because it's one of the hardest problems in ichthyology and has been for a long time, like who's related to whom among the relationships between the carps and minnows and tetras and catfishes and knife fishes. And knife fishes might be the most unfamiliar, but I'll talk to you about them in a minute. But as we transition from sort of a handful of genes to many genes with next generation sequencing, target sequencing, especially ultra conserved elements, which at LSU, you know, we've really been, took hold, especially because of folks like John McCormick and Brant Faircloth. We thought, you know, this must be the magic bullets that we need to figure out the tree of life, right? So with hundreds of genes, we must be able to solve these difficult problems. And, you know, that's not been the case. So I don't want to get into the relationships here, but when we looked at the relationships with Ostreophysins, one of the striking things I found, even though we were using hundreds of genes, we sometimes still had short, deep branches in the tree. And this used to mean it's not enough data, but there's actually a lot of data behind these short branches. It's just a naturally short branch, or appears so. And part of the explanation for that may be a rapid old diversification, but also part of the missorting and incomplete lineage sorting that has happened where, you know, the genes don't explain the true history of the group. But when I think about this, I also think about going back to the earlier part of my talk about recombination, how that can also erase part of our ancestral history. And so these are kind of parallel things at the genomic level, millions of years, and also our own family trees. But there's this difficult question because this group of Ostreophysins broke up on Gondwana multiple times over 100 million years ago. So there remains difficult questions. The knife fishes, which are an Ostreophysin, includes this guy. So this is me on the Amazon. An amateur ichthyologist would have dropped that fish, but not me, you know, professional. So these guys communicate with electricity, just like the pony fishes communicate with light. And they're found throughout the Amazon basin and into Central America. And their relationships were pretty confusing too. So they have different signaling patterns. And with UCEs, my former postdoc, Fernando Aldo, was able to show a very clear, you know, different wave types, pulse wave types, communication types, essentially, of how they use electricity to communicate. And the phylogeny made total sense. But I noticed that there are these short, deep branches, again, back in the end of the tree. And Pam Hart, who I mentioned before on our trip to Thailand, she was looking at North American cave fishes, and her paper just came out in Evolution. And there's just a handful of species, but still short, deep branches. Bill Lute, who's now curator of fishes at LA County Museum, just down the road, 6 hours down the road from you guys, he was looking at this family of marine fishes, Latridae, short, deep branches. So I wondered, why are we getting this pattern? It's not just my lab, by the way, getting these. So I was curious about it, because when we used exon capture for phylogeny of gobies, we didn't get this pattern, but we were filtering the data. We're at this point, we have so much genomic data, so many markers that, we can filter it and sort of not pick and choose, but look at the particular kinds of data we want. And so exons and UCEs are kind of like the Coke and Pepsi of target capture. And, I want to compare them. And again, with Fernando Alda, who's now at UT Chattanooga, we looked at cichlids that had exons and UCEs examined for the same group. And what he found were that the exons are very noisy individually, but better at explaining deeper time divergences, while UCEs were better at younger divergences. But actually the take-home message was from that paper, or the paper he's working on, unstable relationships are hard to solve with all data sets. And this was, not the hubris that we had when target capture came out, right? So for us, we thought lots of fragments, lots of genetic data, genomic level data will solve all our tree of life questions. But this told me that hard questions will remain difficult, at least for the foreseeable future. And so there are limits to our information. This is the Mo data, Mo problems quote from Biggie Smalls. I'm sure he really said that. But to quote another OG, Ed Wilson, don't let anyone tell you that taxonomy and exploration and boots on the ground is finished. So just moving away from that, the papers that I just talked about from our lab, those are really interesting papers for me because I care about the animals, but the papers end up being a lot about the data. And, it brought me back to, Chris wanting me to talk about fieldwork, and the boots on the ground part is still really important. And so I just wanted to sort of end the talk with the section about the role of museums in the age of genomics and the age of COVID, which is how I started it. Bill Lute again had, who's again the LA County curator, who was in my lab, he synonymized these two surgeonfish species, which, 10 years ago, we would have just done with like a ruler, right? We would have used calipers and done measurements and compared specimens, but he used genomics and ecology, how they eat and where they eat, to compare these species to show that they're the same ones. And for me, this is like a model example of modern day museum science that incorporates the old school stuff because he still measured specimens, but also the new school techniques available to everyone. With Janet Buckner, who's a postdoc at LSU, we did this survey of all vertebrate genomes with 30x coverage. So we called 30x good coverage. And we looked at the 1,000 or so genomes that have been done for all vertebrates. And it's only about 10% that have vouchers. And vouchers being a specimen or a record of the source material of what was sequenced. And a lot of us have heard about GenBank having issues with identification. And we think that genomes won't have that problem, but I think they will. And I'll give you an example. So probably one of the most conspicuous animals, not just fishes in the world, is Mola mola, the giant ocean sunfish. And when the genome of this species was done, I mean, it's a really cool paper, but there's no voucher. It was just a blood sample from a specimen that washed up in Florida. Since that paper came out, Mola mola was split into 3 species. So whose genome is it? And this one we can probably figure out, but think about some obscure fish from the Amazon or from Thailand. That won't be so easy to do. So sort of implore everybody to use vouchers. And maybe it should be us museum folks that are the sheriffs telling people to do that. Because it's often us that provide the loans for the specimens, for the DNA that gets sequenced and become something else. And so we need to be biocurators too. And that's a hard thing to ask for curators to do, but when we loan a specimen, what happens to the data afterwards? We're often not acknowledged. What happens to the data after is out of our hands. But what's the tree of life going to be if it's just names on a phylogeny? There's other things that we can hang on that metadata. And maybe it should be us curators keeping track of that. If we add the ecological data and all the other information, it only adds value to the tree of life. And if we add the paleo data, one of my favorite lines from Tracy Heath is she said that if we had all the paleo data, we wouldn't even need to date the tree of life, right? Of course, we would never always have all the fossil evidence, but this will allow us to unzip the tree of life and figure out how over time, species have changed and how fauna have changed. But we need to do that with the metadata. So I'll just end with one last fieldwork photo about me and Diego, my student from Guatemala, while we were in Haiti last year. This is probably the place where I found that we were the most distrusted. They don't have a lot of people coming in collecting fishes. And there's a reason that ichthyologists haven't really been there in the last 50 years. This place, Haiti, which is so beautiful and in lots of places so pristine, but so poorly understood. And so they were very mistrusting of foreigners coming in, even though we were also working with locals. We found that we had to showcase what we were doing right out in our field sites, taking pictures and DNA samples and explaining as we did it. So again, the need for not just finding the truth as scientists, but explaining it so that people can trust us. So I know I'm running out of time, so I'll just go to the conclusions. For people to believe your truth, you need to earn their trust. And I think that's an important thing to keep in mind, especially in the age of COVID, as we're all stuck in our houses. We have to think about how we interact with other people, even though we're missing field work, also how we interact with people in the field. But I think we have an important role, especially the museum folks and the evolutionary biologists, to talk about genomics and COVID and how that role we can play. And then knowing the limits of our data, I think there was a lot of hubris early on, including myself, about what we could do and what we couldn't do. But also in explaining, having knowledge of the explanatory power, not just the limits of our data, but how the stories that we can tell and how we tell them. So I will leave it at that. I'll leave my conclusions up and thank everybody for listening. I know it's not easy to keep track of a virtual talk, but I really appreciate being able to speak to all of you.
Wonderful. Let's unmute and have a round of applause. Are there any questions?
I have one.
Sure, Michael.
It's about the ponyfish.
Yes.
I was curious whether this symbiosis that you described, the bioluminescence, is something that is used all over this clade. Do all fishes use the bacterial symbionts, or some of them have the capacity to generate bioluminescence through their own genes?
In the ponyfishes, the entire family uses the same bacteria. It's the same species, but they're different strains, and that they pick up from the environment. But they all use, and females also luminesce. They all have ventral luminescence. So yeah, all individuals have the bacterial symbiont.
Okay, so the reason I'm asking was because I heard that in anglerfish, I believe, some of them can generate bioluminescence on their own, while others are using bacteria. And I was thinking that the choices between, doing it on your own versus acquiring bacteria from the environment somehow during evolution, perhaps, of the species to do that might be an interesting question. But All right,
Oh, it's a
clarify
great
that.
question. Yeah, and people have looked into it, so they can incorporate the bacterial genome and then co-opt it to produce their own light. There's an anglerfish that the head lure is bacterial based, and the chin lure is their own. So in one individual, you can have both systems. It's so crazy.
Interesting.
I was under the impression that if we had enough data, we could overcome problems like incomplete sorting when it comes to short, deep branches. I gather from you that's not true.
If we had enough, I think if when we talk about genomes, we're never going to use the entire genome, right? Because genomes are different sizes. So the synteny and connecting which part of the genome will always be filtering the data if we use a genome. I think with incomplete lineage sorting, it's a thing that happened, right? So just overcoming it doesn't work with just more data. We just have to recognize it and model it. So modeling that into our phylogenetic work is the best solution. But you're not going to get all of it. And each part of the genome doesn't tell the same story. And it's not the majority either, which is the most disconcerting part. So yeah, it remains a puzzle.
Yeah, I've got a question too. I mean, great talk. And I think you did a great sort of vision of esoteric sort of future museums, right? In terms of better vouchering of genomic specimens. But I guess how do you...
Everywhere I go, I'm going to take Chris and he's going to be my wingman.
Sounds good. I mean, how do you see the sort of interface between museums and the public? And I guess in particular, these countries like Haiti and Tanzania, where I don't even know if there is a local ichthyological collections there. Do you see us starting new collections, or what should we do?
Yeah, there's not only not a collection in Haiti for fishes or vertebrates, there's not an ichthyologist. And so, that's part of why we have to build scientific infrastructure by not just going in there and building a museum. You have to train people. So, when we brought people there was a master's student with us who, slowly learning about the techniques. She was actually more interested in ecology. But just like I recruited people from Guatemala or for Cuba, if you want to really build something there, you have to train the people. And you don't have to expect them to go back either, right? It's just having somebody, a local person who's worked there is so much more important than just going in and helicopter sciencing. So yeah, then you can start talking about building a museum. I've given a fish taxonomy course in Kuwait. I'm like, why are you having an American teach you about your fish? It's because there's no infrastructure here. So it's too bad that there's not really a foundation that can support that kind of international collaboration and building of scientific infrastructure. So it's sort of on us to train those folks and to get them in the network. I see Juan. Hi, nice to see you again.
Hi, Prosanta. This is Juan. Good to see you again too. Thanks for such a fantastic talk. Also, the perspectives on the short branch of Australian fish is fascinating. I'm just curious about one question. The knifefish was hypothesized to be sister group of catfish. But catfish has the fossil record can be traced back to Cretaceous, but knifefish is probably the oldest one. I remember correctly, that was Miocene, sometimes around that time. So there's such a long gap. How do you feel about that? Would that be a ghostly lineage floating around somewhere?
Yeah, thanks for that question, Joanne. Catfishes were distributed, and they still are all over, and there's fossils from Antarctica. So they have these hard, bony spines that fossilize better, which is what's in Antarctica. And knifefishes are soft, they're so you know, so I can imagine that there's, I don't know if I'd call it a ghost lineage that goes way back, but there, you know, we suspect, yeah, they'll be the same age as their sister group, but they're, yeah, there are not that many fossils of them, and that's just part of the fossil record, all those gaps and lack of information. But catfishes are also special in as many fossils that they've left behind. Many more species than the knife fishes.
Yeah, exactly. Right, thank you.
Thank you. Other questions? I can't really see everybody. Maybe I can go back to stop share. Oh, there might be questions in the chat. I'm not sure.
Well, if there are no further questions, Prosanta, on behalf of everyone, thank you so much for the stimulating talk on a wide diversity of topics. And I hope that we can have you come out and visit in person sometime.
Thank you. And I'm saying hi to Carl Spencer, who I just see coming up on that. But yeah, I wish I could have hung out more, but glad to have been here. So thank you very much all for having me.
Thank you. Oh, another round of applause.
Hey, yes, thanks, Prosanta.
Thank you.
Take care.
They're gonna shut it down.